Hi, hello. I'm Shishra Joy, second MSc Biology student, St. Mary's College, Thrissur. Now we are discussing about the topic solute transport mechanism. Transport of solute across the plasma membrane is called solute transport. It has three different types: passive diffusion, facilitated diffusion, and active diffusion. First, about the passive diffusion. It is non-selective process by which any molecule able to dissolve in phospholipid bilayer, they can cross the plasma membrane. Small and hydrophobic molecule can diffuse across the phospholipid at specific rate. Net flow of the molecule is always down their concentration gradient. Example of passive diffusion: the gas, hydrophobic molecules, benzene, small and uncharged molecule water, ethanol. No membrane proteins are involved. No external energy is provided. So in passive diffusion, any molecule can diffuse in the phospholipid bilayer, they can cross across the plasma membrane. The next one is the facilitated diffusion. Movement of molecule in the direction of relatively concentration inside and outside the cell. No external energy is provided. It is different from passive diffusion that transported molecule do not dissolve in lipid bilayer. Polar and charged molecules such as carbohydrate, amino acid, nucleosides and ion can pass. Passage is mediated by protein, two classes of protein mediated by facilitated diffusion: carrier protein and channel protein. First, about the carrier protein, it is specifically bind to molecule on one side of the membrane, then undergo the conformational change that allow the molecule to pass through the membrane and be released on outside. Example, the glucose transport event. This is the glucose transport event. The glucose molecule is attached to the carrier protein. The carrier protein undergo conformation and glucose molecule released. The next one is the channel protein or ion channel. Features are: channel is extremely rapid, 1000 times greater than carrier protein. It is highly selective. They are not permanently open. It is regulated by gates: ligand gate channel, voltage gated channels and chemical gated channels. Patch and clamp technique. Erdwin Neher and Bert Sackman in 1976 study about the ion channel. As Q-daxon, Na plus is 10 times higher in extracellular fluids, K plus is 20 times higher in cystole. So we take a situation of resting potential. In axon is always open to K plus ions and Na plus ions. So Na plus ions move inside the cell and K plus move outside the cell. This is a case of resting potential, leak channel, voltage gated channel and chemically gated channels. In the time of the action potential, there is a potential charge. So voltage gated Na plus and K plus channels open when small initial change in membrane potential. So the Na plus is moved inside the cell. So the membrane potential is minus 50 MV to plus 30 MV. This change is called depolarization. When depolarization happens, the Na plus channel get inactivated. But the K plus channel is open and K plus move outside the cell. Rapid decrease in the membrane potential, say about minus 75 MV, that inactivated K plus channel. So this membrane potential is a resting potential. So this is called the repolarization. Repolarization and depolarization of adjacent region of plasma membrane allow attain action potential to travel down the length of nerve cell axon as electrical signals, resulting in the rapid transmission of nerve impulse over a long distance. This is the action potential, depolarization and repolarization, by which the electrical signals are transmitted through the axons. Then chemical gated channels. In muscles, neurotransmitters releases to presynaptic cell and bind to the receptor of the membrane of the postsynaptic cell, where they can act to open ligand gated ion channels. The third one is the active transport. It is driven by ATP hydrolysis. So first, about ATP hydrolysis. Na plus K plus pump here used energy derived from the ATP hydrolysis is to transport Na plus and K plus against their electrochemical gradient. So 3Na plus bind to the site exposed inside the cell. The binding of Na plus stimulates ATP dependent phosphorylation. Phosphorylation exposes Na plus binding site to the cell surface, lower the binding affinity to Na plus and releases outside. At the same time, 2K plus bind to the high affinity site exposed on the cell surface. The bind of K plus stimulates dephosphorylation of the pump. The pump retains to its original conformation, releases K plus inside the cell. Then next is it is driven by ion channel. Here the molecules are transported against their concentration gradient using energy not from the ATP hydrolysis but from the coupled transport of second molecule in energetically favorable condition. In this situation, Na plus is attached to the opening site. Along with the Na plus, one glucose molecule is attached. They undergo conformational change. Na plus and glucose molecules are released. So along with the Na plus, the glucose molecule can pass through the plasma membrane. Three types of transport: sympod, antipod and unipod. Sympod, transport of two molecules in same direction. Example, 2 Na plus ions and 1 glucose into cell. Antipod, transport of two molecules in the opposite direction. Na plus Ca2 plus. Na plus move into the cell and Ca2 plus move outside the cell. Then unipod, transport only one molecule. Example, facilitated diffusion of the glucose. Thank you.